Here we present our work on 3D fluid flow reconstruction using compact Light-Field PIV. Time-varying fluid flow is a challenging problem, as the fluid is transparent and its flow is invisible. Here we perturb the water with a propeller, but the generated flow is not directly visible with a regular camera. A common practice in experimental fluid dynamics to visualize the fluid flow is to eject tracer particles into the fluid. Then the motion of these particles are assumed to follow the fluid dynamics faithfully, and thus represent the fluid flow. The class of method is called particle image velocimetry. The goal is to track the particles over time and use their motion trajectories to estimate the fluid flows. Faithfully recovering the fluid flow can benefit many scientific research including oceanology, robotics, biology, and fluid dynamics. Existing PIV solutions usually require complex and expensive equipment, and the setups ends up bulky. For example, standard laser-based PIV method uses an ultra-high-speed laser beam to eliminate particles in order to track their motions. In order to recover 3D fluid motion within the entire volume, Tomographic PIV using multiple cameras to capture the particles and resolve their depths in 3D using multi-view stereo. Such multi-view camera systems need to be well calibrated and fully synchronized. More recently, the Rainbow PIV used color to encode particles at different depths in order to recover 3D fluid flow. Although it uses a single camera, the setup requires specialized illumination sources with diffractive optics for color encoding, and the optic system need to be precisely aligned. This paper represents a flexible and low-cost 3D PIV solution. We only use one compact Light-Field camera as the equation devices. No special light sources needed. Our algorithm tracks the dense particle floating in the fluid and reconstruct the 3D flow within the fluid flow volume. A Light-Field camera can be viewed as a single-shot multi-view imaging device. It inserts a microlens array in front of the sensor to emulate an array of pinholes to capture Light-Field records 4D spatial and angular light rays scattered from the particle trajectories. Commercial Light-Field cameras can capture high-resolution Light-Field. We are able to resolve dense particles in 3D fluid volume. Small baseline of the lens rate array further helps recover subtle particle motions at sub-pixel level. Our solution also benefits from the post-capture refocusing capability of Light-Field. In particular, we use focal stack to establish correspondence among particles at different depths. A focal stack of the scene is a sequence of images captured with different focus settings. A Light-Field can be used to produce synthetic focal stack. Here we show Light-Field sub-aperture image could produce a synthetic focal stack, and from far to near. Given a particle Light-Field, we synthesize a focal stack from sub-images by integrating rays from the same focal slice f. Let the IP of f be the intensity of the pixel p at focal slice f. For symmetry analysis, we define an in-focus score kappa here. In order to do the particle depth estimation, we'll formulate a joint optimization function to estimate the disparity in particle area, and using calibrated intrinsic parameters to convert disparity into 3D particle location. The optimization considers traditional color and gradient consistency. We also integrating the focal stack symmetry to estimate the particle depth from depth field. Light-Field helps improving the accuracy and robustness. After we reconstruct 3D particles in each frame, we compare two consecutive frames to estimate the volumetric 3D fluid flow. In particular, we solve this problem under the variational optical flow framework and develop two novel regularization terms, correspondence term and the wedge term. In order to improve accuracy and efficiency, where data is enforced data consistency, in smooth constraint fluid motion to be piecewise smooth. We employ a deformable graph that considers local geometric smooth deformation and rigidity. We use these graph nodes as control points to deform particles instead of computing transformations for individual particles. E-rigid is a rigid regularization term that enforces the local rigidity of a fine transformation. E-smooth enforces the spatial smoothness of nearby nodes. We propose a novel correspondence term for more accurate flow estimation. E-cruise helps in improving the matching accuracy, especially in regions with low particle density. This comparison demonstrates that our correspondence term greatly improved the matching accuracy from 68.5% to 91.5% and hence benefits flow reconstruction. To capture the physical property of the incompressible fluid, we added a divergence-free regularization term EDIF to the optic flow framework. Results show the divergence of the recovered central fluid slice. With or without the divergence constraint, we can see that with EDIF, divergences are closer to the ground truth. In the experiment result, we first evaluate our proposed approach on simulated data. To generate the input data, we position tracer particles randomly within the value. We then show the error with different particle densities, and we use two metrics, average endpoint error and average angular error. Next, we conduct experiments on data generated from the Zhong Hopkins Turbulence dataset. We show that our reconstructed velocity field in XYZ direction. Here is video footage of the slice animation, where ours is middle column, the left is the ground truth, and arrow map animation is on the right. To capture particle images, we use the lateral illumined camera's imaging towards a water tank. We trigger camera under a control board with 10 frames per second. Next, we show our recovered real fluid flow velocity field and past-life reconstructions. The first one is the one vortexing. The second one is the double vortexing. Third one is the random vortexing. We can see that our reconstructions will depict the intended fluid motions and are highly reliable. For more details, please check our paper. Thank you.